Hello friends, this presentation is a part of Enemy ICT project sponsored by MHRD Government of India. Let's start a discussion on microbial fermentation of enzymes. Today we need to discuss microbial fermentation and production of two enzymes, amylases and proteases. Let's start a discussion with amylases. What is amylase? Presence of measure two classes of starch degrading enzyme, that is alpha amylase and glucoamylase, have been identified in the microorganisms. Alpha amylase is also referred as endo 1,4 alpha D gluconohydrolase, which randomly splits the 1,4 alpha D glycosidic linkages between the adjacent glucose units in linear amylose chain. Alternative means of glucoamylase include glucan 1,4-alpha glucosidase, amyloglucosidase, 1,4-alpha-D-glucan, glucohydrolase, which hydrolases single glucose units from the non-reducing ends of amylose and amylopectin in a stepwise manner. Unlike alpha amylase, most glucoamylases are also able to hydrolyze the 1,6-alpha linkages at the branching points of amylopectin, although at a slower rate than 1,4 linkages. Various bacteria and fungi produce amylases, although the enzymes produced from these two sources are not identical. Fungal and bacterial amylases are used commercially in various fields like preparation of sizing agents and removal of starch sizing from woven cloth in textile industries, preparation of starch sizing paste for use in paper coating in paper industries, in liquefaction of heavy starch paste formed during manufacturing of chocolates and corn syrups in processed food industries, in brewing industries, and removal of food spots in the dry cleaning industries. Amylase fermentation. Fungal amylase production utilizes any one of the two processes, that is solid state fermentation or submerged aerated agitated fermentation. Solid state fermentation. It is well known for the production of enzymes by filamentous fungi. As these molds have specific morphology and physiology, they can penetrate and colonize various solid substrates. Various agro-industrial wastes are utilized as substrate in this process that provides both physical support and source of nutrients in the production of numerous fermented products and enzymes. This method offers advantages such as great volumetric productivity, superior product recovery and product characteristics, small capital investment, use of agricultural industrial waste, reduce pollution problem, and a lesser amount of effluent generation and level of catabolite reparation gets reduced. Agricultural waste which are used in enzyme production include starch, wheat bran, rice bran, maize bran, sugarcane, baggage, wheat straw, rice husk, banana waste, tea waste, cassava flour, corn flour, wheat flour, steamed rice, etc. Next is submerged fermentation. Industrial enzymes can be producing using submerged fermentation in which microorganisms are cultivated in liquid nutrient broth. This process involves growth of selected microorganisms in closed containers having a rich fermentation broth of nutrients and a high concentration of oxygen. As microorganisms are growing, they release the desired enzymes into solution. Nowadays, large-scale fermentation technologies are developed which allows significant production of microbial enzymes. Larger fermenters with capacity of up to 1000 cubic meters volumes are used for large-scale fermentation. Common raw materials like maize, sugars and soya are used in the fermentation media. Generally, microorganisms secrete industrial enzymes into the fermentation medium which degrade the carbon and nitrogen sources. There are two common processes, batch-fed and continuous fermentation. To allow growth of the biomass in the batch-fed process, sterilized nutrients are added to the fermenter. But in the continuous process, sterilized liquid nutrients are added into the fermenter at the same proportion as the fermentation broth leaving the system, which lead to steady-state production of enzymes. To optimize the fermentation process, various parameters like pH, temperature, oxygen, depletion and carbon dioxide creation are measured and controlled. First step in harvesting of enzymes from the fermentation broth is removal of insoluble products like microbial cells, which is generally carried out by centrifugation. Most of industrial enzymes are extracellular, which remain in the fermentation medium after removal of the biomass. The biomass is treated with lime to deactivate the microorganisms and stabilize it during storage, and then can be used as a fertilizer. The enzymes remaining in the broth are then concentrated by various methods like evaporation, membrane filtration, or crystallization. Submerged fermentation offers advantages like ease of sterilization as well as process control is simple. The enzyme produced can be constitutive or inducible, which depends on the strain and the culture conditions. Fungal amylases. Fungal amylases can be produced by any of these two fermentation processes. Generally, strains of Aspergillus oryzae are utilized for stationary culture with wheat bran. For this process, the wheat bran spread as relatively thin layer in trays is moistened with water or dilute acid, sterilized and inoculated with spores of a fungus. After growth, the fungus plus bran can be dried at 50 degrees centigrade or less and then ground to serve as an amylase preparation. However, more often the amylase is extracted by water from the wheat bran culture, precipitated from the aqueous solution by the addition of alcohol and dried at 55 degrees centigrade or less. The submerged fermentation for amylase production have recently become economically feasible, and although strains of both Aspergillus niger and Aspergillus oryzae have been studied extensively for their possible use in this process. Fungal amylase are produced by submerged culture method employing the following media. Main component of the media includes cornstarch, cornstip liquor, potassium chloride, sodium hydrogen phosphate, calcium chloride and magnesium chloride. There is a problem of aeration and agitation because of a very high viscosity of the medium due to the presence of mycelial body. Amylases biosynthesis is inhibited when the medium contains glucose. Bacterial amylases. Various bacteria elaborate amylases, but only amylases from Bacillus subtilis and Bacillus diastaticus have been produced on a commercial basis. Bacterial amylases are employed under those conditions in which fungal amylases or amylases from other sources hydrolyze starch less well. For instance, bacterial amylases has an optimum temperature for activity at near about 55 degrees centigrade and the enzyme is relatively heat resistant. As a result, bacterial enzyme finds particular application in situations in which starch hydrolysis must be conducted at higher temperatures. Strains of Bacillus subtilis are specially selected for amylase with high starch liquefying and dextrinizing activity. Subsequently, this enzyme produces comparatively less fermentable sugar when acting on starch. High yields of bacterial amylase are obtained when Bacillus subtilis is grown in stationary culture. Fermentation medium contains a high level of crude protein because high carbohydrate level in the medium stimulates protease production and depress amylase production. The pH of the medium is near neutrally and the fermentation proceeds for approximately 6 days at an incubation temperature of 25 degree to 30 degree centigrade. Bacillus subtilis in this stationary liquid culture produces a heavy surface pellicle growth which apparently is associated with high amylase yield and fresh sterile air is circulated over the pellicle to improve aeration. At harvest, the culture is filtered or centrifuged. The recovered aqueous portion is concentrated by evaporation to yield an amylase concentrate, and salt and an antiseptic are added. As an alternate recovery procedure, the amylase can be precipitated from aqueous solution by the addition of cold acetone, ethanol, isopropanol or ammonium sulfate. Bacillus subtilis amylases can also be produced in highly aerated submerged culture by using a special medium having high content of starch. Bacterial amylases are produced by using following components in media: ground soybean meal, amber, BYF, that is brewer's yeast fraction, distillers dried soluble, enzymatic casein hydrolysate, lactose, magnesium chloride, and antifoam agent. However, the ability to produce amylase is a somewhat unstable characteristic of these organisms, and culture degeneration, which is difficult to detect, may occur during the fermentation. This culture degeneration, however, is not a serious problem with stationary pellicle forming Bacillus subtilis fermentation. Next is microbial fermentation and production of proteases. Proteases, they are the proteolytic enzymes which are produced by various bacteria such as species of Bacillus, Pseudomonas, Clostridium, Proteus, and Serratia, and by fungi such as Aspergillus niger, Aspergillus oryzae, Aspergillus flavus, and Penicillium roqueforti. However, the enzymes associated with these microorganisms are actually mixture of proteinases and peptidases, with the proteinases usually being excreted to the fermentation medium during growth, while the peptidases often are liberated only on autolysis of cells since they are endoenzymes. Complex mixtures of true proteinases and peptidases are usually called proteases. At present, the proteinases are of more commercial interest than are the peptidases. Among the commercial applications of proteolytic enzymes is the bedding of hides in the leather industry. Here the enzymes bring about changes in the hides to provide a finer grain and texture, greater peelability, and better general quality. Proteolytic enzymes also are employed in the textile industry to remove proteinaceous sizing and in the silk industry to liberate the silk fibers from the naturally occurring proteinaceous material in which they are embedded. In addition, these enzymes are employed in the tenderizing of meat, and they also are the active ingredient in spot remover preparations for removing food spots in the dry cleaning industry. The fungal proteases present a wider pH activity range than do animal or bacterial proteases, and to a certain extent this results in a wider range of use for the fungal proteases. These are two types of proteases: alkaline serine protease and acid proteases. Alkaline serine proteases are mainly produced by Bacillus licheniformis by submerged culture method. Acid proteases are mostly produced by fungi by either semi-solid culture or submerged culture method, depending upon the fungal species employed. Fungal proteases. Various fungi produce protease enzyme in good yield, and the commercial production of fungal protease has utilized Aspergillus flavus, Aspergillus venti, Aspergillus oryzae, Mucor delamer, Mucor mihi, and Amylomyces roxy. The fungus is usually grown on wheat bran, although other media are sometimes employed under fermentation conditions similar to those for amylase production. At sporulation, the various fungal proteolytic enzymes are present in the medium, and the proteases are recovered by procedures similar to those for mold amylases. The optimum temperature of the fermentation is 30 degrees centigrade and requires 3 days for completion. Submerged fermentation procedures for fungal protease production have been studied, and the indications are that such fermentations may become commercially feasible. Mucor mihi is used to produce acid protease by submerged culture method using media having components like starch, soybean meal, ground barley, calcium carbonate. The optimum temperature of the fermentation is 30 degrees centigrade but requires 7 days for completion. Bacterial proteases. Bacterial protease production again utilizes strains of Bacillus subtilis, and the fermentation conditions are similar to those for amylase production by this organism. However, the Bacillus subtilis strains are specially selected for high protease activity and not for amylase activity. As stated previously, a high carbohydrate content medium is utilized to stimulate protease activity and depress amylase production, although the final product does not contain some amylase activity. may contain some amylase activity. The fermentation is incubated 3 to 5 days at 37 degrees centigrade in pans containing a shallow layer of fermentation medium, and the harvest procedure is similar to that for bacterial amylase, except that concentration of the broth is carried out at reduced pressure and at temperature of less than 40 degrees centigrade in order to protect the enzyme from denaturation. Alkaline serine proteases are obtained from Bacillus licheniformis by submerged culture method using media having components like starch hydrolysate, soybean meal, casein, and sodium hydrogen phosphate. Thank you. So by that we have completed discussion of fermentation of amylase and protease. We welcome your questions and feedback. Please visit us at our website www.elearnmicrobiology.co. Hello friends, this presentation is a part of NME ICT project sponsored by MHRD Government of India. Let's start a discussion on chromatography. In many fermentation processes, chromatographic techniques are used for isolation and purification of relatively low concentrations of metabolic products. Chromatography is generally used for the separation of mixtures based on the differences in their shape, size, structure, and or composition. In chromatography, components of the mixture to be separated are distributed between stationary phase and mobile phase. Components to be separated are carried by the fluid or gaseous phase is called mobile phase, which moves in a definite direction and carries them through a structure holding another material, which is termed as stationary phase. Some components interact tightly with the support and can move slowly through the support than other components which interact weakly. Different components of the mixture move at different speeds which finally get separated. Components are separated because of their differential partitioning between two phases, that is the mobile and the stationary phase. For example, in liquid chromatography, as liquid is passed through a column, it allows passage and separation of different solutes. Depending on the mechanism by which the solutes may be differentially held in a column, the techniques can be grouped as adsorption chromatography, ion exchange chromatography, gel permeation chromatography, affinity chromatography, reverse phase chromatography, and high performance liquid chromatography. First, adsorption chromatography. This technique involves weak van der Waals bonds to bind the solute to the solid phase. To pack columns, different inorganic and organic materials can be used. Inorganic adsorbents utilized here includes active carbon, aluminium oxide, aluminium hydroxide, magnesium oxide, silica gel, etc. For example, active carbon in concentration of 0.25 to 0.5% is used to remove pigments and other impurities from penicillin containing solvents. Organic microporous resin may be used for same purpose. The first synthetic organic microporous adsorbent, the Amber Light XAD resins, were produced by Rom and Haas in 1965. These resins do not have any ionic functional groups but have surface polarities which differ from non-polar to highly polar. Second is ion exchange chromatography. Ion exchange chromatography is used for the separation of ions and polar molecules like large proteins, small nucleotides, and amino acids on the basis of their charge. Molecules of acidic, basic, and neutral nature can be easily separated by this technique. In this separation, matrix-bound charged groups reversibly adsorb oppositely charged sample molecules. Diffusion of the molecule through the matrix of the exchanger is dependent upon the degree of cross-linkages of the exchanger and the ionic strength of the buffer. This diffusion process can control the rate of the whole ion exchange process. However, desorption of the bound ion is achieved by change in pH or changing salt concentration or by adding competitive counter ions, since it tends to disrupt the electrostatic interaction of the matrix with the bound molecule. Ion exchange chromatography is perhaps the most frequently used chromatographic technique for separation of proteins according to their net charges. Types of ion exchange chromatography. Mainly there are two: cation exchange chromatography and anion exchange chromatography. In cation exchange chromatography, the stationary phase contains a negatively charged functional group which holds positively charged cations, while in anion exchange chromatography, the stationary phase contains a positively charged functional group which holds negatively charged anions. Cationic ion exchange resins normally contain acidic active groups like sulfonic acid, carboxylic acid, etc. Carboxymethyl cellulose is a common cation exchange resin. Positively charged components like certain proteins will bind to the resin. Then, by passage of buffers of increasing ionic strength or pH, deposited solutes are successively washed away. Anionic ion exchange resins normally contain a secondary amine, quaternary amine, or quaternary ammonium active group. Diethyl aminoethyl cellulose is a common anion exchange resin. Weak acid cation exchange resins like amber light IRC50 can be used in the isolation and purification of antibiotics. For example, to recover the streptomycin, sodium form of amberlite IRC50 is used. On the Onto the column, by displacement of sodium ions, the streptomycin is adsorbed. The resin bed is now rinsed with water and eluted by hydrochloric acid to release the bound streptomycin. Reaction is already given. A slow flow is used to ensure the highest recovery of streptomycin using the smallest volume of eluent. In one step, the antibiotic has been both purified and concentrated, maybe more than 100-fold. The resin column is regenerated to the sodium form by passing an adequate volume of NaOH slowly through the column and rinsing with distilled water to remove excess sodium ions. Commercially, it is not economic to regenerate the resin completely, therefore, the capacity will be reduced. In practice, the filtered broth is taken through two columns in series while a third is being eluted and regenerated. When the first column is saturated, it is isolated for elution and regeneration while the third column is brought into operation. Ion exchange chromatography may be combined with HPLC in certain cases. Next is gel permeation chromatography. This technique also recognized as size exclusion and gel filtration chromatography. The separation of the components in the sample mixture is based on their molecular weight, shape and size. Generally, it is used to determine the molecular weight of the molecule. Gel filtration is also an important preparative technique in the purification of proteins, polysaccharides and nucleic acids. The stationary phase consists of porous glass granules or gel particles which is in equilibrium with mobile phase. The smaller molecules having lower molecular weight can move into the gel faster than the larger ones and penetrate the pores of the gel to a greater degree. As a result of this, the larger molecules which are completely excluded from the pores of the gel will be eluted first when elution is started. Intermediate sized molecule elute in the middle of a large and small molecules as they can fit inside few but not all of the pores in the bits. However, molecules of small size are completely included. They get distributed between the mobile phase inside and outside the molecular sieve and elute last in a gel filtration separation. Various gels are available like cross-linked dextran, that is Cefadex and cefacryl, and cross-linked agarose, that is saffrose, with various pore sizes depending on the functional range required. One early industrial application, although on a relatively small scale, was the purification of vaccines. This technique harvests a pure portion of vaccine which is then concentrated 10 times by removing the eluent buffer by pressure dialysis. Next is affinity chromatography. Affinity chromatography is used for separation and purification of most biological molecules on the basis of their functional or chemical structure. This technique depends on the highly specific interactions between pairs of biological materials such as enzyme substrate, enzyme inhibitor, antigen antibody, etc. Separation is based on the binding affinity of biomolecules with their respective biologically active ligands. This ligand is bonded covalently or by physical absorption to the solid matrix, which interacts with its analyte as a reversible complex. Eluent is then passed through the column to release desired molecule from the reversible complex in highly purified and concentrated form. The major components required for an affinity chromatography procedure are bead matrix, ligand, and a solution containing the substrate to be isolated. A wash to elute the non-bound impurities in the solution and a final wash to elute the bound substrate from its ligand. For efficient separation of product, the pore size and location of the ligand must be corresponding to the size of the product. Coupling procedures have been developed using cyanogen bromide and related compounds for matrices of gels and beads. Four polymers which are often used for matrix materials are agarose, cellulose, dextrose, and polyacrylamide. Most common support used for the coupling of amino ligands is agarose gel activated with cyanogen bromide. An effective alternative to gel supports found in affinity chromatography is silica-based solid phases. Purification may be several thousand folds with the good recovery of active material. The method can, however, be quite costly and time-consuming, and alternate affinity methods such as affinity cross-flow filtration, affinity precipitation, and affinity partitioning may offer some advantages. Affinity chromatography was used initially in protein isolation and purification, particularly enzymes. Since then, many other large-scale applications have been developed for enzyme inhibitors, antibodies, interferon, and recombinant proteins, and on a smaller scale for nucleic acids, cell organelles, and whole cells. In the scale-up of affinity chromatographic process, bed height limits the superficial velocity of the liquid. Thus, scale-up requires an increase in bed diameter or adsorption capacity. Next is reverse phase chromatography. Most liquid chromatography, that is normal phase chromatography, uses a silica or alumina resins as solid support stationary phase, which is hydrophilic in nature. Thus, the hydrophilic molecules in the mobile phase, which have strong affinity for the stationary phase, tend to adsorb to the column. At the same time, the hydrophobic molecules are eluted by passing through the column. Opposite to that, in reverse phase chromatography, hydrophobic stationary phase are used where solid support is covalently bonded to alkyl chains. Generally, 8 to 18 carbons containing alkyl groupings are used. A hydrophobic stationary phase has a stronger affinity for hydrophobic compounds. Thus, the hydrophobic molecules in the mobile phase tend to bind to the column while the hydrophilic molecules pass through the column. To allow elution of hydrophobic molecules from the column, an organic solvent of decreasing polarity is used, which reduces hydrophobic interactions. This allows the separation of molecules according to their hydrophobicity. Wide variety of molecules can be separated by reverse phase chromatography, but it is not generally used for separation of proteins because sometimes proteins get denatured by the organic solvents used in the technique. Sometimes this chromatography can be combined with other chromatographic techniques for the separation of specific molecules. Now last is HPLC, that is high performance liquid chromatography. It has also termed as high pressure liquid chromatography. HPLC uses smaller, more rigid and more uniform beads. Such type of beads can also be produced by improving the nature of column packing material for a range of chromatographic technique, that is gel permeation and ion exchange chromatography. This allows packing of columns with minimum spaces between the beads, which results in minimizing peak enlargement of eluted species. Because of that, resolving power of HPLC will be always superior at the time of separation of mixtures. So it is a high resolution column chromatographic technique. HPLC uses better quality media in terms of their selectivity and physical properties for the solid phase through which the mobile phase passes. The stationary phase uses in HPLC contains high surface area upon unit volume. Even size and shape of that is resistant to mechanical and chemical damage. However, it leads to high pressure necessities and cost. In terms of principle, LC and HPLC work the same way, but HPLC is much superior in terms of the speed, efficiency, sensitivity, and ease of operation. This may be acceptable for analytical work but not for preparative separations. Thus, to reduce operating and capital cost in preparative separations by HPLC, some criteria are often compromised, like instead of small, larger stationary phase particles are used. But at the same time, for the separation of very high value products, significant HPLC columns containing analytical media have been used. Affinity techniques can be combined with HPLC to get benefit of both, that is, the selectivity of the affinity chromatography and the speed and resolving power of the HPLC. So by that we have completed discussion of each type of chromatography. Thank you. We welcome your questions and feedback. Please visit us at our website www.elearnmicrobiology.com. Hello friends, this presentation is a part of NME ICT project sponsored by MHRD Government of India. Let's start our discussion with introduction to cell line culture and vaccine production. Currently, maximum vaccine manufacturing methods are based on classical egg-based technology. Almost all leading vaccine producing companies are working hard to develop new and novel methods for preparation of vaccines. Most efforts they have made till now were based on improvement of currently available licensed egg-based vaccines. All these companies are running to increase production capabilities and mechanize maximum manual steps in egg-based vaccine technology to satisfy the demands of the next seasonal campaign as well as to create prototypes of pandemic vaccines for clinical trials. All these manufacturers are functioning with international agencies like World Health Organization, the National Institute of Health, and European Medicines Agency on the improvement, licensing, and making of pandemic vaccines on a global scale. There are various advantages of cell culture technologies over egg-based methods, like the need for embryonated chicken eggs from managed biosecure groups get eliminated, the upstream and downstream processes get automated, the potential for contamination by viable and non-viable particulates get reduced, the time needed for the organization of egg supplies get eliminated, production would be faster, initial purity would be higher, they could increase seasonal vaccine supplies when numerous strain variants are essential. They would significantly raise global supplies on pandemic influenza vaccines. Cell culture based vaccine production. This is the robust and reliable technology which could be a best practical alternate for the vaccine producing pharmaceutical industries. First, the desired virus is propagated and harvested, and then downstream processing parameters like purification, filling, and packaging of the vaccine are required, which are same as the currently available egg-based methodologies and pharmaceutical methodologies. This method uses cell line. The process can begin once a cell line is infected with the desired seed virus in a fermenter. The most critical need is the seed virus. The media used for cell line proliferation are not susceptible to infectious virus strain as embryonated chicken eggs are. This process is appropriate for large-scale vaccine production and the process parameters can be cost effective. Typically, Cell culture vaccine production technology can be run in batch size of practical scale, which is sufficient to provide vaccine quantities for expected demand. However, till date, no vaccines have been certified using this technology, but Chiron has already submitted mock-up reports to European Union regulatory authorities for review, approval and a license application for a cell culture based avian influenza vaccine. Which are the steps which we need to follow to produce this vaccine? To begin bulk production, the virus should first cultivated in a fermenter having many process parameters to control temperature, pH, dissolved oxygen and other factors. For mass cultivation of cells, mainly two methods are recognized in the industry such as microcarrier cultures and free cell suspension cultures. In microcarrier cultures, the cells are first attached to microbeads. In the presence of nutrients, the cells grow and proliferate, covering the beads evenly. As high surface area to volume ratio is provided by microbeads, it leads to high cell densities. Once a bead is covered, the cells are displaced, detached, and allowed to retach to reach another round of cell growth on the surface of the bead. In case of free cell suspension cultures, higher volume of media are required to achieve the same results because the cell line proliferates only while cells are growing freely in the nutrient medium. Though the scaling up of the system is easier and there is no limit to the volume. Cell line selection. The cell line used to propagate the virus in huge quantities must be quick and capable in cultivating the desired virus. It must be appropriate for a wide variety of viral strains. It is necessary that the cell line be capable to grow in a chemically defined synthetic medium which does not have animal derived constituents. It should also be accessible for industrial processes. Similarly important, if the cell line has not been approved earlier by regulatory agencies, the necessities for licensing should be known and legalized. Serum based media. There are some disadvantages with serum based media. Acceptance standards for serum can differ as much as 20%, which could lead to batch variations during fermentation. Possibilities of contamination of serum with adventitious agents. It can be challenging to isolate the target protein from the serum protein during purification if the target protein is attached to a serum protein. For large-scale cell culture use, a serum-based medium is not always available. Synthetic media. There are some advantages with synthetic serum-free media. It is much better defined media than serum-based media. The possible source of infectious agents has been removed. There is much less batch-to-batch variability than for serum-based media. The purification of the desired protein is easier as it requires fewer steps and a lesser cost. Synthetic media contain readily available non-animal derived components that are relatively easy to store. Lastly, shortages are unlikely. Removal of residual DNA. During selection of a cell line for vaccine production, one most critical step is the removal of residual DNA from the end product. Generally, regulatory agencies are providing necessary guidance for continuous cell lines. Continuous cell lines must not have tolerance for adventitious agents or other contaminants. The removal of DNA must be much more systematic. Testing models have been defined to measure potential risk and to confirm safe use by the public. There are three strict regulatory requirements for certifying a new cell line for use as a substrate in cell line culture preparation. There must be records to support the complete elimination of the cells from the final product as well as to confirm that the cell line does not carry any transforming agent into the final product. Documentation must be there which confirm complete absence of genetic material from the cell line in the final product that may otherwise cause tumors in animal models. Finally, records must indicate the removal of infectious and or oncogenic agents from the final product. The strict regulatory requirements are proposed to protect the public from changes in the immune system caused by extraneous DNA. A complete description of the cell line is compulsory to meet licensing requirement in any country. To express the influenza virus, currently following cell lines are used: per-C6 EBXTM, it is a stem cell line derived from chicken embryos. Vero, it is kidney cell from the African green monkey. In MDCK, in this median Darby Kenny kidney cells used in Chiron. MDCK cells are famous to produce huge amounts of virus and need easy downstream purification. Though this cell line has not yet been approved by regulatory agencies, it would be a significant biochemical engineering achievement if an influenza vaccine applicant used cell culture manufacturing that contains MDCK cell growing in suspension in a synthetic medium. Vero is newly licensed with regulatory organizations but does not express huge quantities of virus. Vaccine manufacturing and formulation. Suppose a cell line can express large quantities of virus and regulatory agencies will support it. One has to think whether the cell line can develop with the industrial means, whether we can grow it in a fermenter, and which type of culture, that is a free cell suspension culture or microcarrier, can be used. Both these methods have engineering challenges. The cell line must be grown in a nutrient medium which may be either synthetic, that is serum free, or a complex having animal derived components like protein or serum. The risk factor is less with synthetic media if they stimulate the growth of the cell line. But the presence of serum creates a safety hazard as well as introduces a potential source of contamination. So the use of synthetic media has increased significantly. During vaccine formulation and manufacturing by cell culture process, first the decision of type of vaccine, that is whether a whole virus vaccine, a split vaccine or a subunit vaccine should be finalized, then qualified and validated. Most of egg-based vaccine are split or subunit vaccines, but a vaccine for pandemic influenza is most probably to be a whole virus vaccine. First, cell line propagation initiates with the small-scale proliferation of seed cells after thawing. Then cells are inoculated to the fermenter vessel containing specific nutrient media. Once a predetermined cell density reaches in cell line, the virus is inoculated and starts to proliferate in the cell line. Then the virus is harvested approximately after 3 days. Then after the virus is released into the supernatant and the cellular debris is separated by centrifugation. All these processes occur in a clean, closed environment. Subsequently, the complete virus can be purified, fragmented and ultra-purified as a subunit. Initially, ultrafiltration is used, which is frequently followed by treatment with beta-propiolactone that deactivate the virus. Ultimate splitting of the virus is followed by ultracentrifugation. This ultra-purification process is fundamentally similar to the egg-based vaccine ultra-purification procedure. Because of that, the resulting purified subunit vaccine is same as egg-based vaccine in composition. After that, remaining most important task is to complete the licensing process. Phase 3 clinical trials in Europe have revealed equal safety and immunogenicity of cell culture influenza vaccine strains and traditional egg-based vaccines. For both formulations, the protective line is well beyond the lowest limit. In the United States, phase 1 and 2 studies were accomplished in 2005. Adjuvants. Adjuvants are constituents which are added to vaccines to increase antibody production and the immune response of the receiver, or to decline the amount of antigen, that is dose size, required in the vaccine. The last option is the almost effective way to increase vaccine manufacturing capacity at globally. Till date, the only two adjuvants, that is aluminium compounds and M59, have met regulatory standards for safety. Aluminium compounds have been used safely for many years, and M59 is licensed in most European countries. Since commercial operations initiated, more than 25 million doses with M59 are administered. So one can understand that cell culture based influenza vaccine production has many benefits as compared to egg based vaccine production and should be licensed by regulatory organizations in near future. Adjuvants are not only effective but also increase global vaccine manufacturing capacities by antigen sparing. Thank you. We welcome your questions and feedback. Please visit us at our website www.elearnmicrobiology.com.